phases.
Okay, thanks, Sarah. If we hand on to Ali, please.
Hi, morning everybody. So my name's Ali Miles. I'm also working in the Environment Agency. So I started on this incident back in October to support Sarah and her team, but I was at the national level, focusing around monitoring and assessment side of the Environment Agency.
Thanks, Ali. We move next to Chris, please.
Yeah, hi everyone, I'm Chris Evans. I'm a fish health. My day job is on in Wales, but I was involved in this case at an early stage looking at whether disease was a possible cause, but we moved on to supporting DEFRA in December time, so I was leading the coordination of CFAS science, reviewing the case and seeing where we could take things forward and different avenues that hadn't been explored.
Thanks, Chris. If we can move to the MMO team, please.
Hello, I'm Chris Proud. I'm the Senior Marine Officer at Scarborough. So I work in fisheries enforcement and maintaining a sustainable fishery in this area. And obviously I cover the majority of the area where this wash-up has occurred. And this is my colleague.
Yeah, I'm Pat Gray. I'm the Principal Marine Officer for the North East team. So I work with Chris and we're responsible for the regulation of licensed activities in the area.
Thanks, guys. We can move now to the IFCA, to David, please.
Hi, everybody. Hopefully, I think everybody should know me by now, but I'm David McCandless, the Chief Officer at North Eastern IFCA. Our IFCA area covers the Humber, North East Lincolnshire, up to the Tyne, South Tyneside. So obviously the impacts of this event have fallen squarely within our IFCA district. Thank you.
Thanks, and to Tim, please.
Yes, thanks. Hi, Tim Smith, Senior Scientist, North Eastern IFCA, supporting.
Thanks, Tim. Okay, and Gabriella, please.
Hi, I'm Gabriella. I work in Core DEFRA as part of the business management team, and I assist on the EPL cell.
Thanks, Gabriella. So, Joe, if I could hand over to you to introduce, please. So, Joe, you might be on mute. Sorry. Thanks, Joe. Thanks, guys. Next up from the industry, I've got Paul W up there. Paul, you're on mute. Paul, you're still on mute. No, can't hear anything. Okay, we've got next is David. David, I think you're the...
That noise is David, I think.
I think the noise
Yes.
is David. David, could you dial that in, please? Okay, we'll see if that helps. Paul, I see you're off mute now.
Hello, well, are you getting that?
Yeah, getting you loud and clear.
Hello, I'm Paul Woodfield. We work out of our pool in like inside 6 mile on Sally 65.
Great, thanks, Paul. Sally? Sally, you're on mute.
Can you hear me now?
Yeah, loud and clear.
Yeah, sorry about that. Yeah, I'm the North East Coordinator of British Stars Marine Life Rescue. I sort of kind of look after the marine mammal medics on this area. And I've been dealing with a lot of the emaciated seal pups that have been washing up since the die-off and still are, to be honest. So I'm sort of just interested to see where this is going.
Thanks, Sally. We've got Tim Dear-Jones, please.
Okay, hello. Can you hear me?
Yeah, loud and clear.
Good, right. Yeah, I'm Tim Dear-Jones, marine pollution researcher and consultant. Been working in the field for 30 odd years. Done quite a bit on dredging over the years, and working with W. Whitby Fishers Conservation Group. So, hi.
Good morning. Thank you very much. We've got Christopher, please. Christopher, if you're talking, you're on mute. Hi, Christopher.
Tim, you're not talking. Are you talking to me, or is there another Christopher on the line? I can't see another one.
Oh, it must
Sorry.
be you, Chris. Sorry.
Yeah, no worries. All right, sorry. I couldn't see the list. It's me. I've introduced myself. I won't do it again.
And sorry, and then we've got a name, Widowfield. I'm not sure who that is, please.
Yeah, I've already said it once.
Oh, sorry. Okay, you're signed in twice, Paul. Thanks. Cool. Okay. We haven't had another sign in, so we'll see if they join us in a while. Okay. If we start the meeting over then, so. Okay, so just a reminder to all that obviously we're working towards the same goal here, and that's to try and understand what's happened over the recent months and last year, maybe mostly through October and December, and just to emphasise the work that DEFRA has done to look at this, but also to appreciate the input from Tim Dear-Jones's report and the kind of the review he did of the work as well. So thank you for that. So just in light of this, just remember that obviously with the call, if you're able to put your hand up either on the electronic version on screen, or just wave at me on screen, and I'll call you into the conversation if you want to make a point as we go through. So thank you for that. Okay, so as a quick overview of where we are, I'm sure we all know where we are, but as a quick overview, there's been an extensive range of testing which has been undertaken over the last several months by DEFRA Group and the agencies working with us. The EA, MMO, CFAS have all looked at potential causes, including chemical contamination, dredging, discharges, and potential disease issues. These causes were ruled out, and more detailed information as on why this is the case will be explained today, specifically focusing around the pyridine question. The North East NIFCA's report on healthy crabs and lobsters is now are reporting that there's now been caught in the region, and I'd appreciate some feedback from those on the call on that at the moment when we go through later on. But we will continue to monitor the situation, and the investigation has not been closed. There's no conclusive explanation that we can find at the moment for the incident, but the evidence is still suggesting around the harmful algal bloom to be the most likely cause of this incident. So if we move on to the further details of the work that's been undertaken by the group and the findings that we've had, we'll start to move through that. So if we move to the first agenda item, which is on pyridine, and I think Ali, you're going to lead us on the pyridine, please.
Yes, will do. And Adam, if you can put my slides up onto the screen, that'd be very useful. Thanks. So, as I say, my name's Ali Miles. I work for the Environment Agency, and the Environment Agency led on this incident investigation for the first couple of months in October and November of last year. We've done a whole range of work, but I'm gathering from the feedback from people that what we're actually wanting to know more about is the chemical side of the investigation. So that, and specifically pyridine. And so that is what this presentation is about. This first slide was just really to set the scene about the chemicals and how we operate under incident investigation around chemicals, and then I'm going to specifically go into pyridine. But when we start off any incident where we believe chemicals were involved, there are certain things that we're looking for in terms of large toxic pollution of incidents. And these are the steps that we've kind of followed within this investigation. So the first thing, of course, is we're looking for contaminants or chemicals of concern in water samples, in biological samples, in sediment samples. But we are looking for levels that sit outside historically what we see. It's really important to keep in mind, if we analyse or anyone analyses an environmental sample, you will find chemicals. The key thing about this was whether it's actually the cause of the mortalities that we saw. So the first thing we look for is actual chemicals in the environment. The second one, if we have a point source of chemical, a toxic chemical, into these kind of waters, you all know these are very exposed waters, it's mixed coastal waters. Thank you. We would expect to see the sort of dilution mitigation. So in other words, as the chemical goes into the seawater, it dilutes. So over time and space, you expect to see a reduction in any chemical impact. And the third key thing that we really see is a whole range of species obviously affected. We know right from the start that the impact of this was very obvious within crabs and lobsters in particular. And so that was a sort of another key factor of this investigation. So that's what we look for, regardless what the incident is, that's what we're looking for in terms of large toxic pollution incidents. So how I've set up this presentation, I'm just going to go through an overview of pyridine and our steps through the pyridine investigation. I'm then going to break it down a bit more, look at the crab results, the measurements we made in crabs. I saw we were requested to explain a bit more about how we analyse that, so I've put together 2 or 3 slides just to say how the analysis actually happens and why we keep referring to these as indicative results, just to try and explain that a bit more. Going to go through what we found in the water, what we found in the sediment, what we found in blue mussels. So that's all our environmental compartments really that we've monitored in. Then talk a bit about why we believe dilution is such an important factor in this investigation. And then I'll just recap and bring together that weight of evidence to say why we have concluded what we have. I realise there's an awful lot to take in. So what I suggest I do is go through the overview, go through the crab tissues and the methodology, and then just have a bit of a pause there to see whether people have any specific questions about that methodology and the biological sample results before moving into the second part. I'm actually working off a phone, so I'm going to find it very hard to see if people are waving at me or putting their hand up. So if I pause after that analytical methodology discussion, perhaps Tim or Adam can also highlight which questions we've got, what are relevant to come in there. Otherwise, we'll take a note of them and I'll respond to them at the end of the presentation. And hopefully that works for everybody. So if I can have the second slide, please. I do apologise for the amount of writing on this slides, but I know everybody is really keen to understand all the different steps. So I've tried to put as much in as I can. So we're now going to just focus on pyridine. And this is what we went through in terms of our investigation around this incident, specifically on pyridine. When we had the first crab results, the first indicative values coming out from the crab tissues, and I'll explain a lot more about why those are only indicative at the moment. And at this stage, so this was early on, at this stage, the impact was still fairly localised around the Tees and around the Tees Bay area. So we had to presume under our instant response, that pyridine was the cause. It's important to realise at this point, we didn't actually even know it was specifically pyridine, and we didn't know about concentrations, but we reacted to that signal that we had seen in the crabs. This immediately sets off a range of different steps that we undertake for incident. So we would start looking on literature in terms of what's known about the ecotoxicology. So what's known about how these how pyridine acts in the environment, what generally we find with pyridine, and specifically we started to try and look to say, what do we know about pyridine in crabs and lobsters? And at this point, we were looking very much about this impact on the crabs and lobsters and the very specific behaviour in terms of the twitching and lethargic behaviour of the crabs and lobsters. So the next stage was, we again presumed it was pyridine without being able to confirm it. We presumed it was, and we go looking to find a potential source of that. What could be driving that? And this ranged a whole range of different activities, including taking formal water discharge sample. But the issue was we couldn't find significant pyridine in the other compartments of the environment. We couldn't find it in water and in sediment. So we couldn't identify a significant source of pyridine. As we were doing this, the impacted area, and the time was passing, the impacted area was increasing. So suddenly we don't have this dilution mitigation that we would be looking for. Because of this, and because we'd been looking for a point source, like a point of discharge of the pyridine, it means that contaminant source becomes increasingly improbable, because we're not seeing it in the environment and we're not finding any dilution mitigation of the impacts. So then the thought process went to, actually, we need comparison crabs from outside the area. We've never analysed for pyridine in crab tissue before. We don't know what we would normally expect. So we looked for crabs from outside the known impacted area in order to analyse those as well. And we analysed pyridine in water, in sediment, we did it in blue mussels, we did it in impacted crabs and in comparison crabs. And basically, we couldn't find pyridine in the water. So we started thinking, could the pyridine that we were seeing in the analysis actually be linked to biological processes in the crab tissue? So in other words, is it a consequence of this incident rather than a cause? Could it be from the stress and the decomposition of the crabs as they were dying? So that's the kind of stepwise thoughts that we went through within this investigation. And if I pass on to the next slide now, I'm going to focus on the crabs themselves, and this is what started this line of inquiry. So right from the start, we needed to find chemical, if it was going to be a chemical pollution incident, but say we presumed it was at the very start, and look for a signal that we could follow up. And we thought would be best to take some of the dying crabs from the impacted area and try and analyse those to see if we could find a signal, a chemical signal in the tissue that we could then follow back to source. So this map shows, the red dots show where we initially took impacted crabs from for the analysis. We also took some comparison crabs from South Shields and St Mary's Lighthouse. So in the region area, but outside the impacted area that was known. And those comparison crabs appeared healthy when they were sampled. We also went further afield. We took 2 comparison crab samples that came out of the Norfolk Wash area, so still on the east coast, but outside the impact area. And then we even looked at a sample from a different regional sea by asking for a sample out of Cornwall, out of Penzance. So those are the comparison crabs that we got. And again, I want to emphasize, just because we find chemicals in the tissue, it does not mean that they are the cause of the deaths of the crabs and lobsters. Next slide, please. So what I've done here is I have ranked the samples in terms of indicative concentrations of pyridine. And I'm going to get to the indicative bit and about the method discussion on how we get this. But these are all the crab samples that were taken in this early stage of the investigation. The samples in green are comparison crabs. They have been taken outside of any impacted area that we are aware of and appeared to be healthy crabs. What you can see from this is there's a range of indicative values, but if we look at the Norfolk Wash two comparison crabs we took for there, you can see one has got an indicative value of 3 and one has got an indicative value of 195. So one of the samples from the comparison crabs is actually within the levels that we are seeing in the impacted crabs. If we hadn't have seen any pyridine in the comparison crabs, we would have followed up even more on pyridine in the impacted crabs. But this finding that it's actually there in unimpacted crabs led us again to think, is this a consequence of, or is this part of the biological processes within crab tissues? It's not something we've ever measured before, so we have no idea. So next slide, please. So the next 2 or 3 slides, can't remember exactly, were in response to, I think, Joe Redfern, you requested more information about how we actually do the analysis. And I'm not an analytical chemist, but I'm hoping that this will give us all an indication of how this is done, how this works. So we use something, the GCMS screening technique, analytical technique, to actually look at what concentration, what chemicals sit in the crab tissue analysis. It's really important to understand this is a method the Environment Agency uses for screening water samples, and it's accredited for looking at water samples. So it was developed to look at a whole range of substances, including pyridine, but in water, and that is really critical for understanding these results. However, in response to the seriousness of this incident, we asked our lab service to adapt the method to look for chemical signals in biology, in biological samples, in other words, the crabs, and in sediments. So we have a method, and we've used part of it that's well established, for example, the solvent extraction, that I'll talk about in a second. But again, responding to the fact that this hasn't been tested in crab tissues. We did this under incident investigation to provide us with as much information as possible about potential chemical leads. We don't actually know how this method really behaves when we analyse biological tissues. And again, we don't have a baseline for what we would find for pyridine in crab tissue. When you analyse a material using this method, what you'll see that diagram, and that diagram is just illustrative. It's not of crab tissue. I just tried to pick a spectrum off the internet in order for you to understand what comes out of the instrument. Basically, you put your sample through the instrument and you will see this kind of spectrum come out. And all these different peaks are different chemicals that you are finding in that tissue or in that water sample. Next slide, please. So we have this GCMS screening method that's developed for water. And really the question was for this, how far could we push it to look for crab tissues? As I say, its method is accredited, but it's done for water and it's not done for crab tissue. When you measure pyridine, it's kind of in 2 parts. The first part is we have to identify that what we're seeing, the peak that we're looking at in that spectrum of peaks, is actually pyridine. And the second part is to determine how much pyridine is present. And basically what you do with your samples, your crab tissue, you're liquidizing it, mashing it up and putting it in a solvent, in this case dichloromethane. and the chemicals move from the tissue out into the solvent. And that solvent is then injected into the instrument, and that's where you get the spectrum from. It looks at the chemical signatures you find in that solvent. So you liquidize your crab tissue, you pull the chemicals from the crab tissue into the solvent, put the solvent into the machine. The instrument then for pyridine, the pyridine molecule is basically broken apart in the machine by firing electrons at it. Pyridine itself will always break apart in exactly the same way. So you can compare it to like having a fingerprint. We have a library, our labs have a library of these different peaks and the different way that this breaks apart, and we compare what we see in the sample with what we have in the library. The difficulty with moving into a different, like in tissue samples, is you could almost consider it now that it's a bit of a dirty fingerprint. Crab tissue has a lot of fat content, so you start seeing slight interference. But what we can say is if we see that fragment, if we see that signal in the peaks, we know that we've got pyridine. And actually following up, at the time we didn't, but the Environment Agency purchased a standard to run in the machine to double check that the peaks we were seeing were actually pyridine. So we do know that what we saw from this method is now pyridine. At the time, when we started doing the full investigation, we hadn't got that confirmation, but we acted on it anyway. If I can go to the next slide, please. So the second part of this whole thing is measuring the amount of chemical that we have. When the instrument sees pyridine, the instrument responds. So the more pyridine you've got present, the more the instrument responds. And we report this as a number, and we call it a concentration. And that's what you'll have seen in the data sets under the concentration. And we use the number milligrams of pyridine of the chemical in every kilogram of crab. And that's how you'll see the numbers reported. The difficulty for us in this investigation, while we've screened water samples for pyridine before, and there's a lot of analytical methodology in order to calibrate that, we've never done it in crabs or sediment. So what we had to do was use our understanding of how the water works and basically extrapolate it, use it to try to estimate concentrations in the crab tissue. It's unlikely to be very accurate, but we don't have the data to confirm this. But it gives us a clue, it gives us a signal of which we can follow up in the other matrices. And I think it's also important to state at this time, when we first got the samples out from the crab tissue, our lab only reported concentrations of less than 1,000 milligrams per kilogram. They didn't want to differentiate numbers below that value because we didn't have this calibration, this knowledge about the crab tissue. But those of us involved in the investigation needed to have something more. We needed to know relative values. So we asked the lab to put those numbers or to give us those numbers, even though they couldn't confirm it. So we could consider high, low, medium concentrations, and it would give us further indications in which we could investigate further. So what this means is, although we know the instrument measured pyridine, there are things we don't know, and that's what would require a lot more testing. But we don't know whether the solvent it removed, in other words, the signal that we're seeing, whether all the pyridine was actually removed from the crab. When we put the tissue in the solvent, does it suck out all the chemical into the solvent? This is what we call recovery or bias. We don't know if we've recovered all the chemicals that sit in the crab tissue. We also don't know how consistently it does it. That's what we call our precision. So if we go to the next slide, please. When we look at these samples, and again, we'll go back to the Norfolk Wash sample, we've got one at 3 and one at 195. We don't know if the levels of pyridine in those two crabs was exactly the same, or it was just the pyridine levels in crabs were different. because if you take the same sample and analyze it many times, you might get different results. That hasn't been done for this kind of methodology with this tissue. This was, remember, is an investigation, incident investigation. We were working at pace and we were using this to signpost us to things to investigate in other matrices. All this information basically meant that we were more and more convinced that the pyridine was actually linked to biological processes in the crab tissue rather than an environmental cause for the mortalities. As I say, I've gone through that speed. That is quite a lot of information I've thrown at people. I don't know if we want to pause now if people have got any specific questions about that method and about the results we saw. Yep.
Okay, so we've got Joe's got a hand up, Joe.
I can respond quickly to that, Joe, but it might also be worth bringing those points into the conversation at the end when we've seen all the different slides. But we were looking at such a significant impact here, we would expect to see these values within the crabs. They were enough for us to signpost to look for the pyridine in the other compartments. And this is why I need to come back to this perhaps at the very end, because when I've given you the supporting evidence on the water and the sediments and why we'd expect to see it in those environmental compartments as well, then we need to consider whether actually measuring it in crabs any further is worthwhile, or whether it's actually it's the wrong line of investigation to take in terms of understanding what's driving this incident. So Joe, is that okay if I can just come back to that? If Adam, perhaps you can just make a note of that to make sure we do cover it at that point. Yeah, so we've been trying to look into the ecotoxicology, sort of the levels of that, where you would predict to get an effect of different chemicals within crab and tissue. There's not a huge amount of information out there, to be honest, especially around crabs and lobsters in this way, but we do have some indicative levels that we can do. So perhaps again, Adam, can you make a note and I'll make sure we come back to that at the end. Yeah.
Okay. And then we've
Sorry,
got
Joe, on my phone, I can only see a sort of thumb coming in from one side. I've got your colleague fully on screen, but yeah.
It's okay. So it's Tim Dear Jones, please.
Hi. Thank you for that, Alison, so far. Interesting presentation. I was just interested in your comparative crab samples. Did you take account of the fact that, for instance, St. Mary's Light appears to me to be quite close to Tynemouth? Did you take account of the possibility that Tyne estuary itself carries high levels of PAH, amongst which pyridine seems to be included and listed? And can you chart or give some information as to Norfolk Wash 1 and 3? Are they two separate crabs from two separate sample sites, or are they two crabs from the same sample site? In which case the same question pertains, because if one of those crab samples was closer to an area which might be a source of pyridine, like an industrial or a port area, than the other, that might have relevance on the results.
Yep. So just again, I probably need to bring these back in on the wider conversation, but
Okay.
no, what we went for in terms of the comparison crabs was just getting crab samples outside of the known impacted area with crabs that weren't displaying any behaviour of what we were seeing in the incident as well. So we're looking for the chemicals driving this specific incident. So we didn't think about what other sources might be around wherever the comparison crabs. We were just trying to see where the pyridine is found in other biological material, be it naturally or anthropogenically. So they weren't specifically chosen. They weren't control crabs. They were comparison crabs outside of the known area of impact. Yeah.
Right. Yeah.
I can't confirm the difference between the two Norfolk wash crabs. They were two separate crabs that were analysed, but I don't know about their handling. I don't know exactly where they were sourced from. So they were provided through the Eastern Ithaca, just as this indicative, do we find pyridine elsewhere in samples around the coast? I mean, a key thing with this on the pyridine levels might also be, we don't know the stress or what period of dyeing, what point in the dyeing process the crabs were sampled. So we've no idea whether it was a freshly exposed sample in those impacted area, or whether that was a crab that had been exposed over several tidal cycles.
Yeah.
And that was when. So those are unknowns for us. As I say, I can come back into that later if we still feel there's questions to answer on that.
No, so far so good. Thank you.
Yeah. So
Thank you.
what I'll do is I'll go through the next slides, if that's okay with you, just to build up this sort of weight of evidence that we're talking about. And then I'll come back to different slides if people have got certain questions about specific things. If that works for everybody, Adam, perhaps we could move on to slide 9, please.
I think Sally's got a question.
Sorry, I've got Sally's
Oh,
hand up.
sorry.
Hi, it was just having worked in analytical chemistry in my early days. You talk about the methods used to extract the chemical from the crab, and you've kind of admitted that these are not reliable results, in that you don't actually know whether the processes you are using are actually effective. So I kind of like blows my mind, having been in quality assurance, to think that we're relying upon anything when we can't actually say the methodology used is reliable, and that actually we are extracting everything that we're looking for. It just seems to me that, we should be doing more work to actually prove that what we're doing can be relied upon.
So Sally, and this is, yeah, it is very difficult, but remember, this is incident investigation.
Yeah.
So we used all the validated methods we have for the other analysis, but we're now looking, these techniques, these screening techniques that we use in water will screen for hundreds of samples at any one time. And we were looking for a needle in a haystack of what could be this cause. And that was our priority, to understand what we were looking at. So we were willing, under incident, we wouldn't report it if we were having to report formal results into classifications that we do for water quality. We wouldn't use this method, but we were looking to try and understand at pace what could be driving it. So they were only to be used as a signal. In other words, for us to follow up with validated methods. It was just trying to get a clue to what might be. So I think we have to separate out what we would use for formally reporting samples versus what clues we are looking for in order to act fast and also follow up in the different compartments.
So have we now got a process in place where you're happy to present that as a formal conclusion, or are we still looking at vague analysis? Have we got a process where we can say that this is reliable?
So, Sally, can I come back to that?
Yeah.
Again, I feel I'm just coming
No,
back.
no,
But can I add that into the end? Because
yeah,
this is where it comes into this weight of evidence. So the crab tissue analysis, we need to consider that as trying to get a signpost, trying to get a clue to what we can then formally look at, because otherwise we don't have any clues. We didn't know what we were looking for. So I'll come back to that one as well, and we'll just check off that we've covered everything. But yeah, I wish I had a fully validated method, but you know how many chemicals are out there. You know the analytical challenge for looking for them. So was there anyone else?
Okay, Ali, can we move on now to the next part of your presentation?
So one part then was these, that's the crab tissues. That's were the signals that we were seeing in the crab tissues. If I can move to the next slide, please. So this is what the Environment Agency does more as sort of its bread and butter, almost to say, is looking at chemical signals in the water itself. And what we're doing is trying to look to see what different chemicals or what different signals we're seeing in the water around this incident compared to historically what we see in the area. We know we will see chemicals in any water samples, and so we have to try and split off that difference between what's causing an incident and what's normally found. And this is where we do have fully accredited screening methods. They're still semi-quantitative, but these have gone through a lot of analytical development. And there's two different ones that you'll have seen results from, the LCMS and the GCMS. But that's the sort of numbers of samples these techniques in water have been validated for. And I know I have shared across with you the sort of database for all the chemicals these methods can detect. So if I could go to the next slide, please. Again, apologies, there's just a lot of information, and I'm assuming we will share the slides afterwards so you can see more in detail about this. But what I was trying to say with pyridine is we have included these GCMS water screening methods since about 2012 within our surveillance programme. So we have rolling programmes to look at water quality in different parts of estuaries and coasts. And what you'll see is we know we see pyridine occasionally in water samples in the Tees. For awareness, we also see occasional detects of pyridine in other saline waters around the coast, for example, in Southampton Water in the Orwell estuary. So finding pyridine in the water isn't a new thing in terms of these waters. But actually, in this investigation, we had the boat survey, and it moved up from Flamborough Head up to the Tees area. So taking samples at different locations in the impacted and outside the impacted area. In over 20 water samples, we only had 3 with positive detects, in other words, that we saw a peak in those sort of spectrums, and they were at the levels of detection. And as of those 3 samples, one of the positive detects for pyridine was actually outside the impacted area, and it was south of Flamborough Head. Pyridine is soluble in water and it's highly mobile. Of this extent, we would expect to see the pyridine in the water sample because it moves easily between sediments and between water column, and it's soluble in water. The other problem with this is pyridine in an oxic environment, so where it's got oxygen in terms of when it's in the top layer of sediments or it's in the water column itself, it would degrade very quickly. So we're not seeing the pyridine in the water compartments where we would expect to see it should this have been driving the event. Next slide, please. We also touched on the fact that we had looked for pyridine in sediments, and we did this initially at the beginning, I think it was the 8th of October. Samples were taken by Sarah's team off of Brand Sands in Tees for an area that we knew was taking the impact, and we did the GCMS screening on 3 surface sediment samples from that area. It's got method limitations similar to the crab tissue, but we were looking for these indicative results. We were looking for that signal in order to signpost it as a lead for what we needed to investigate. These were surface sediment scrapes only, because the investigation was focused on newly deposited or newly disturbed sediment. We weren't looking to find historically what might be buried in the deep layers of the sediment in anoxic conditions, because that wasn't driving this incident. We needed a new signal. It would have been in the surface sediments if it had been something that was driving this incident. Of those samples that we saw, only one sample showed pyridine present, and this was again at its limits of detection. Just to note, when we did our boat surveys, we did take sediment samples as we went, and these have all been frozen for future analysis if needed. But with the pyridine, the signal just isn't there in the sediment on that location. Just if I can move to the next, so that was the sediments. That's what we're drawing from the sediments. We also did a blue mussel analysis on the GCMS screen. It has the same issues as crab tissue analysis. It's indicative. We were looking for signposts. Pyridine was detected, and we got an indicative level of 11 within those tissue samples. This is like, again, beginning of October, these samples were taken. These mussel samples, this is a site that we measure historically. So we've gone back to 2003, looking at the data. We don't measure pyridine regularly, as I've just been explaining about the methods, but we do measure pHs within blue mussels. And it's quite important. Pyridine is not a PAH. It's not a polycyclic aromatic hydrocarbon. They are different toxic effects. But I know we have talked a bit, and pyrene has been mentioned, so I just thought I'd put this up to show what the historic trend shows, and pyrene is one of these, but this is the PAHs in the Ts. What you'll see is the 2021 sample, that's the one on the far right, doesn't show that much of an increase, especially if you put it against the 2014 peak that we saw in PAHs in the Ts, that we think was linked to the North Sea surge and the disturbance that was happening at that time. And in that 2014 event, there were no mortalities that we were aware of in the biological community. So historically, and this isn't just the Ts, this happens in various locations around the coast, we see sediment disturbance, we see remobilization, but what we don't see is this kind of impact on the biological communities. So I say that graph is the pHs, but I know that's been brought up. But again, pyridine is not a PAH as such. And pyrene is not a derivative of pyridine. They are structurally different, and then they have different ecotoxins. So that's all the different samples that we have, which were giving us these signals to understand where the pyridine should be followed up or not. The last key, if I can go to the next slide, please. The last key point in terms of what we were looking at is about the dilution effects. Now, very kindly, colleagues in CFAS have allowed us to show this imagery, but it is really there to illustrate the point. As we were going through considering different chemicals in this incident, we were doing sort of mass balance calculations. How much chemical would you need to impact an area over this time and space? But this is a very good visualization. And this is a hypothetical situation, so this isn't a true situation, but we said, what if we took a point source? How far would that chemical get in terms of impacting an area? And what you can see is 3 different scenarios here, releasing 100,000, 10,000 litres, in this case of pyridine. And we look at this in different chemicals. Any chemical of concern, we would go through this kind of indication. And to say, okay, if this had happened, how big an area it would impact? And what you can see is the area of impact under these scenarios remains fairly low. Just so happens in this example, we said the source would be in the area of a dredge disposal, but that same scenario would work if that was off a discharge from an industry or anything. What it's there to illustrate is the dilution, the huge dilution effect that this mass of seawater would have on any point source of chemical release. So if I can go to the next slide, please. So this is the recap of why we moved away from thinking that pyridine was actually the cause of these mortalities. We have pyridine in crab tissue, we know that, but we find it in both impacted and comparison crabs. Even taking the indicative method that we've got there, you know, not reading too much into the concentrations, why are we finding this in the comparison crabs if this is related to this incident? So pyridine sits in healthy crabs. We've got no obvious signal of pyridine in the surface sediments. These are disturbed sediments that are newly settled within this incident. There is no significant pyridine signal in the water samples around this incident. And this is above sort of what we know historically we can find within waters. I've just put this in brackets because, again, pyridine is not the same as pyridine. But, you know, the pHs, even if we looked at pHs, because we did look at those earlier in the investigation, they're still sitting within historical levels that we've seen in the Ts, where there was no follow-on mortality in the biological community. So it doesn't sit outside of what we'd expect to see. They have got this huge dilution factor in exposed, well-mixed coastal waters. And this was an event going on over a large area in time and space. And what we would need to see, we would be seeing levels of chemical concentrations if we had a chemical that was doing this kind of impact. So that's our thinking. You're probably relieved to know that's the last slide in this set. I hope that's been clear to communicate. I'm very willing to share these slides if people would find it useful or take follow-up questions on it. But that's the logic. That's the logic that we've gone through in the Environment Agency. We find a signal, we follow up, and then does it all align? In which case, you know, then that becomes likely that that is the issue. What we've got in this case is clues and signals from the chemistry, which we then just can't match up with what we're finding with the rest of the picture. And that is why we said it's highly improbable that this is a chemical cause for this incident. And I'll sort of stop there, and then, you know, if people want to revisit any of the questions or some bits aren't clear, I say I'm happy to take that now, or if people individually want to just get a bit more clarity. On there's certain parts I can take away and get more experts back from our Ecotox, our chemical assessment unit, or from our analytical chemists within our laboratory service to just try and clarify that. But that's the narrative around pyridine as seen from the DEFRA group. And I hope you found that helpful. But
okay. Thanks, Ali. Are there any questions from anybody at the moment on this, please? Tim?
Can I just ask, can on behalf of the Whitby group, can I ask for a copy of your presentation, please? Unfortunately, I haven't been recording it, so some of your additional detail over and above what you've got on your slides I've missed, but I've got a plethora of notes here which would take me another hour to go through with you, so perhaps I can contact you later on with an additional list of written questions, and I'm particularly interested in being able to nail down precise positioning of the sources of the comparison crab samples. because what you have there doesn't really seem to accord very well with the control sample from Penzance, which is a town that I know very well, and I know there is no industry there likely to be producing pyridine or related chemicals with the CHN basic molecular structure, although in different amounts. So I would be really interested in nailing that down, and also perhaps some idea of the kind of ground that those crabs were taken from. Were they taken from rocks, or were they taken from sediment-rich areas? And also one other query that you may be able to answer now about Brown Sands sediment sampling. Did anybody do any granulometry on that? Because Brown Sands certainly, although I haven't visited and walked on it, looking at it on various satellite photography and Google Map, it looks like sand rather than a sediment, a fine sediment source. And I would be interested if anybody had done any granular work on that to just define the size of the sediments that were involved there.
Yep, so I would have to double check on that. We usually analyse a less than 63 micron fraction, but I will check because, I say, under incident mode, we will be looking wider than our usual standard practices. In terms of the crabs, I think our key thing for this investigation, because we're not really interested in whether we get pyridine in crabs from around the coast. What for us was the key thing was to say, those crabs were healthy and they've got pyridine in them. So that to us wasn't signalling that the mortalities in the northeast were being driven by pyridine. I will try to do what I can to follow up on that. I'm not saying that, but that's the difference on the incident. It's like, we know we're going to find chemicals when we look, and we haven't really looked in crab tissue before, but would be surprised if we didn't, because we see them in every other biological matrix we analyze for. What the difference is if there was pyridine in those crabs, why weren't they showing the behaviour that the crabs on the northeast were? That's the critical. That's why we call them comparison crabs. We're not saying they're control. They're not cleaner in any way. They're just outside the area of this incident. And all our focus was on trying to understand this incident rather than the wider chemical picture around the coast. But I say I will do what I can to find that out. Happy to share the presentation. I will try and write notes underneath each slide to recap what I've been saying, but also happy if anybody from the association wants to follow up those points with us to get a bit of clarity.
Right, thank you. And just one final thing: do you have good academic data sources for the understood? Because I mean, in my report, I did note that there was a consensual agreement that pyridine did occur naturally in the environment, but all the data I came upon said that it occurred at very low levels, and I'm not so sure that the St. Mary's lighthouse figure, for instance, reflects what I would call a very low level, whereas the Penzance control crabs might accord with that. So if you had a data source for, you know, the normal consensually understood background levels in marine species, I would be very interested to see that.
It is very difficult. There isn't a huge amount of data out there. A lot of what we're looking for in terms of pyridine toxicology comes from things like the regulation that we have on the hazard of different chemicals, also for the European Chemistry Agency to understand what we know about this. But some of the literature, and this is why we have gone, as well as not finding it in all the other compartments, a big thing that we've looked at is trying to understand, would it be as a decomposition? You know, these crabs were impacted. They were obviously stressed. They were dying crabs. You know, could pyridine be formed there? And again, for crabs and lobsters, there's very little information out there. But there are indications in terms of decomposition process from things, humans, pigs, etc., where we know the pyridine, that we know this group of chemicals starts to get formed in this decomposition. Again, we don't know for crabs and lobsters. We are trying to get the wider picture of what we understand about these chemicals. But again, I will put everything we've got, you know, it's being made available to you, and we can add in those sources as well.
Yeah, thank you. I'll share with you the difficulty in finding any data at all on these subjects. It's very hard indeed, wasn't it? Thank you, Alison.
Yeah.
Okay, thank you. Are there any more questions for Ali at this moment, please?
And Tim, could I just go back to Joe and Sally, I think, to say whether that has answered their questions? I'm very aware that I've put them to the end of the discussion. So, Joe, does this give you what you need at this time? Right. Yeah. Yeah. Okay.
Yeah, that's. No, sorry, that was my mistake. That was we have closed investigation. What we're now looking at is the next steps. So how we move forward with this. So the active investigation is closed, but we're moving on to next steps now. So monitoring and yeah, how we work on the situation going forward. Sally, were you happy with the rest from Ali, or is there something else you need to pick up?
Well, I don't think my question was really answered as to we still don't know we have a method that actually is reliable and works. And as Joe said as well, I am concerned that sampling was only a scrape when we know the dredging went down 10 metres. So we don't necessarily looking at an impact that's happened recently. We could be looking at historical stuff that's accumulated over time, and also the sampling of the crabs. You know, for me, it's all very well to blame a biological process, but unless you look at living crabs as well to prove that actually there were samples of 430 in the living crabs that were still on their last legs. You know, I just think, for me, the investigation isn't complete, and it's a long way from complete. If I was running it, there's a lot of things I'd have been doing differently, but I'm not, and I appreciate how much work you've done, but I have got a plethora of questions, and I will forward them on, and thank you for the opportunity.
Okay, thanks, Sally. Are there any other points at all that anyone wants to raise right now on the pyridine question? No, okay, thanks. So if we move now on to Chris, the CFAS findings, please.
Yep. Hi, everyone. So I'm going to be sharing my screen. Hopefully I can get this right. Okay, hopefully you can all see that. That's my presentation. So yeah, as I said at the beginning, I'm Chris, Fish Health Inspector at CFAS, but I took over coordination from the CFAS point of view of the investigation into this case in December time, just reviewing the science and looking at possible other avenues, as I said, that we could look at as to what was the cause of this case. So just as a quick overview to the presentation, I wanted to start just by going through what our responsibilities were and generally the testing that was carried out by CFAS. I won't dwell on this too much because I appreciate that obviously we've given presentations before to Whitby Fishermen's Association in December. I'm not going to rehash that kind of old ground and go into detail. We also don't have very much time. And then I'll concentrate on this harmful algal bloom theory, going through some of the satellite data, the phytoplankton species that were identified in the region, and then also onto marine biotoxins, algal toxin results that we got from some of our testing. And then just at the very end, just touch on this more recent report of dead lobsters that were caught in the impacted zone a couple of weeks ago by a fisherman fishing in the area. So in terms of CFAS's responsibilities, I think it's worth just saying that, I mean, from our point of view, the only statutory responsibility that we had was in the Fish Health Inspectorate to investigate whether there was a listed or emerging aquatic animal disease in this case. At a very early stage, you know, we were seeing what looked like an impact on largely decapod crustacea. So, you know, it was a very real consideration that this could be an aquatic animal disease. Like I said, I'm not going to go into the details here because I think most people are well aware of what we have found and what we didn't find. But essentially, we said that a disease was very unlikely to be the cause of this event through you know, extensive sampling in the area. We also it is important to note that CFAS do provide routine sampling and technical advice to the Marine Management Organisation, who issue marine licences for activities such as dredging. So we do perform routine tests and assessments on sediments ahead of dredging events, and all of these reports are made publicly available on gov.uk. You can find those reports. So we also assisted the early part of the EA investigation by carrying out heavy metals analysis in crab tissues. Again, we didn't find anything here that would lead us to believe that heavy metals were behind this mass mortality event. We also assisted by carrying out samples for naturally occurring harmful marine biotoxins, and obviously I'll go into more detail on that later on in the presentation. So as I said, from December 2021, CFAS were tasked with exploring other possible explanations and supporting DEFRA and the wider multi-agency investigation in trying to kind of drive this case forward and seeing what else we could do. So if I just go into some detail on this theory that on the harmful algal bloom. Now, I think it's first of all important to just raise here the fact that we started looking at this because other avenues were looking less likely. We could see, I've put this graph up, hopefully you can see it, this temperature graph. We can see that sea temperatures were higher than normal in and around this time, the time of the event, around September. I have highlighted here just this red box, which is kind of the area of interest, the timings, which shows quite a very rapid drop, which is unusual for this time of year, this kind of rapid drop in temperature. Now, there's not much I can take from it other than just to say that crabs have died before in quite large numbers, but that has been as a result of going to very cold temperatures. So we're not saying that here, but this could be of significance, particularly if there are algal blooms in the area. They're likely to crash quite rapidly because of this temperature drop. So I thought it was important to first of all recognise that temperatures were unusually unseasonably high around this time. The data from EA routine phytoplankton samples give us a picture of what's happening in the region, but again, it's important to note that this takes place on a monthly basis, so we don't have that full systematic record of harmful algal bloom presence throughout September on the Yorkshire coast. But what we do know is that harmful algal blooms, including Karenia miyamotoi, which I'll talk about in a bit more detail, were occurring in early September in high densities in the northeast region. So around Beadnell Bay, there was a large bloom in the early part of September, 385,000 cells per litre. So we know this is happening. We know this is happening around these sort of temperature marks. The other thing to note here is that there are multiple other phytoplankton species identified and cell densities measured, and this included various other forms of harmful species, including Dinophysis and cyanobacteria in the northeast region. So moving on to the satellite data, so we started to look at some satellite data, and these are developed basically from algorithms that look at the chlorophyll concentration in the water, and we can see across multiple platforms that there appeared to be, quite a significant algal bloom in these regions. So these platforms are available online. They use various different algorithms to correct for things like sediment in the water. Some don't do that so well. But also what we had here, this bottom image is from the Plymouth Marine Lab, which was indicating a harmful algal bloom in the region at the time. I think, it's important to also recognise that as part of the EA sampling in response to the system, we did actually identify Karenia miyamotoi at depth on Teesside. A relatively low numbers, really, to be honest. But again, this was 1.5 months after the onset of the mortality this sample was taken. So we know that the species was present in the area. And then things started to kind of fit together here, because similar cases were happening in the US, where lobsters were dying in pots. They were getting significant problems in Cape Cod, and it was all related to this particular species of algae. Now, in their case, it was looking like a significant oxygen crash at depth, and it was interesting. I spoke to some of the researchers out there and kind of how they approached the case. The other thing that's important to note here is that Karenia mikomotori has also been linked to aquatic animal health impacts, in the sense that the actual algae can cause mechanical damage on the gills. So it's been shown to cause damage to gills in fish and crustacea previously. So this is all kind of evidence that we're building here, and it's, that helps us with the case, I think. But I do think, as Tim's rightly points out in his report, it is really important to recognise that, of course, there are uncertainties with any satellite data. It's never going to be definitive. I think, there are valid questions that Tim raises in his report, and it includes, we don't know from satellite data, we don't know definitively what species we're looking at, as well as the bloom density as well. But I think from the data, what we can be convinced of at least is that we are looking at a bloom of some sort, an autumnal bloom at least, but totally recognise that there are uncertainties with the satellite data. But there is additional information here, which is, is useful, and that's on harmful algal blooms and in terms of the sort of toxins that we're seeing. So, moving on to the marine biotoxins. Early on in the investigation, we did take samples for marine neurotoxins. So this was domainic acid and saxitoxins. And we didn't find them present in crabs at levels that would cause a concern. So that was good. We saw that and we ruled that out effectively. However, we did take further tissue samples, and these were provided to us from the IFCA. They were from dead crab and lobsters that were washed up on the beaches on the 8th of October, so at the onset of this event. They were sent to CFAS, and we screened them for a much broader range of algal toxins in light of this new information about potential algal blooms in the area from the satellite data. We sampled the hepatopancreas, and we did find other toxins, specifically the diuretic shellfish toxins, so DSP, and these are caused by okadaic acid and dinophysis toxin 2. Now, 90% of the samples collected from the region as part of the investigation contained these DSP toxins, and some of them were at very high concentrations in a couple of those crabs. So it's, again, I think it's important that we use the word theory here. We don't know what the significance of this is in the context of the mortality event. It's not fully understood what kind of impact these toxins could have. But the literature does say in certain areas that, first of all, DSP toxins, there is one paper that does link it to neurotoxic effects. And if we go back to what Ali was saying about also the kind of clinical signs that we were seeing in the crabs at the time, we were seeing this sort of twitching behaviour. Now, again, I'm not saying it is, but if DSP are causing neurotoxic effects, they can impact upon the neurons in animals. That's a possibility. The other thing here is that we know that okadaic acid's mode of action is to effectively interfere with enzymatic processes within animals. Now, in crabs, it's again, this is a theory, but it's possible that these enzymatic processes could have been impacted in the crabs that would meant they would be compromised effectively, and opening them up to impacts and death from other actions. So, for example, algal bloom crashing, we don't know about the oxygen crash. We don't have that data. We don't have oxygen data at depth around that time when the mortality event happened. But we also don't know about the kind of interactions of algal species on the gills of the animals. So this toxin data, it is significant to a point, but again, I mean, it's not very satisfactory, I know, but we just don't know what the significance is here. But with the weight of evidence, we're looking at a potential here for it to have an effect on crab health, crab and lobster health, crustacean health. So just lastly, just touching on this recent report of dead lobsters caught in the area, I do think it was important to kind of mention this because we talked about kind of reopening the case and looking at it off the back of this. Now, I did speak to the fisherman who caught them, and he said that they were two dead lobsters caught in the pots, but it was amongst an otherwise healthy catch, and it was catching lobsters and crab in other areas, albeit yes, it reported lower levels, which is, you know, obviously important and significant to consider. We received the lobsters in the lab, and we started processing them for the algal toxins information. So we looked at algal toxins in this material. Toxins were at trace levels, so we weren't finding the same levels that we were at the early part of the investigation. But what was significant here was that we could see the gills of the lobster were heavily parasitized by what's called the lobster louse, this Nicothia stasi. Now, that's not unusual to find around the coastline. So I think it's important to say that whilst we found this ectoparasite in these lobsters, we didn't find it at the early part of the investigation. So it doesn't explain why we've seen this mortality, but it does explain perhaps why we've seen these two lobsters dead in the pot a couple of weeks ago, because it would affect the respiratory function of the animal. So I did want to mention that because I do think that was important, because we sort of talked about, you know, whether this was an ongoing case or not off the back of that incident itself. So yeah, I feel like that's probably a bit of a rush job, but I realise we're running out of time anyway, so that's probably a good thing. I am more than happy to take questions, and hopefully I can answer them. Thank you.
Okay, thanks, Chris. Any questions at all, please, on to Chris on this one? Tim?
Tim, you're on mute.
Hello. Hi, Chris. Thanks for that presentation. It was very, very interesting. Given the finding of DSP in crustacean samples, do you think that may have compromised crustaceans in respect of those crustaceans who had what looked like elevated body concentrations of pyridine?
So yeah, I think I don't know if I can talk about pyridine necessarily. Obviously, Ali's gone over the thoughts there, but what I would say, and like I said, I appreciate this is kind of an unsatisfactory position to be in, because we just don't know what the effect of these DSP toxins would have on the crab and lobster. Now, I've been speaking to the researchers in the lab here, and they're talking about this being like PhDs worth of work to really kind of understand, probably more than that, to be honest with you, to understand what impact these toxins could have on their physiology and whether they are able to adapt to say, for example, conditions which would be hypoxic. Now, there is a train of thought and evidence there that it would affect their ability to be able to adapt to certain environmental conditions and pressures, but this is a really complicated picture. And like I said right at the beginning, that graph, that temperature drop as well, I mean, we don't know whether that could be significant as well, and adding to the pressures that these animals are under. So it's a very complicated picture, but honestly, I can't answer your question, Tim, I'm afraid, but it is something that, you know, I think researchers could absolutely look at in the future, because these toxins are of interest for sure.
Thank you.
Okay, thanks. Any other questions for Chris at all, please?
Sally?
Bye.
You're on mute, Sally.
Sorry. Yeah, I've read that many reports. I'm getting a bit confused now as to where I've read it. But I did read in one report that it was believed the algal bloom would have broken up by the 6th of October. And I was just wondering if that was the case, and we're talking about an oxygen crash, would that still have potentially happened, you know, several days afterwards if this bloom had broken up? And with regard to the oxygen crash, how would that present itself within the dead crabs? I mean, are we able to actually, and I know you do a post-mortem on a person, you can tell whether they've died from asphyxiation. So is that possible to test for in these crab samples to see that there was a definitive cause of death, or is it just an hypothesis which we've got no real proof for?
Yeah, again, I'm sorry, it's a hypothesis, but I think the oxygen crash, I mean, it's based on the satellite data, and as I think, like I said, it's important to recognise that satellite data. You know, we can't. If I was to hang my hat on that, I wouldn't be doing a very good job. I think what we're looking at is the other evidence here as well, particularly the toxins data, like I said, and the actual identification of species. Now, I think the other interesting point here is this species that was found, and potentially the mechanical damage on the gills. Now, when we took our histopathology analysis for the disease, what we were finding is potential sort of degradation of the gills. But again, we can't really see. It looked like that could have been just as a normal kind of background level, to be honest. Is what we sort of see around the country. But again, we don't know whether that impact, the mechanical impact of an algal bloom, could have that same effect on crab, killing them. So yeah, again, probably haven't answered your question very well there, but I don't think there's a way that you can really tell sort of post-mortem whether the impacts have been caused by an oxygen crash. It's not like you get with fish that kind of have their mouths gaping open, gasping for oxygen. You get this kind of effect on the gills, but yeah.
Thank you.
Okay, thanks. Any other questions at all at the moment? Okay, in terms of time, is it okay if we just move on to next steps, and then we'll loop back to questions, if that's okay at the end? So Adam, if you could take us through next steps, please.
Thanks, Tim. Yeah, so just in terms of the next steps, there's 3 elements which will be taking place. So the first of which is what's called a lessons learned exercise. So typically in situations of this nature and in projects in general, you'll look to conduct an exercise to review what's worked well and the kinds of things that you can improve on. So we'll be conducting internal exercise across the whole multi-agency to look at you know, all the different elements of this and have those discussions. So on top of that, agencies on this call, CFAS and EA, are also conducting their own lessons learned exercise. So we'll look to kind of review that, and then once we've done that, we'll be hopefully using that information to kind of review the existing protocols and the existing ways we go about doing things, and using that information to improve in future. On top of that, I'm just going to hand over to David from the North East IFCA. There's obviously work going on in relation to the monitoring and potential recovery of stock. So I'll just hand over to Dave for that one.
Yeah, thanks, Adam. Hopefully everybody can hear me. In terms of our focus now, and I think all industry colleagues, like we met at the end of February really to discuss next steps. In terms of the IFCA, the focus very much for us now, and we've heard extensive presentations from, you know, the lead experts, what I would class as lead experts in the country that have been investigating this extensive investigations. So for me, our focus now is trying to understand what impacts this event has had on the stock and where the stock is sitting currently. And there's a lot of challenges with that. It's not the best time of year to get a clear, and I think our fishing representatives would support that are on the call. It is not the best time of year to get effective data and to actually understand where stock health is sitting. The fisheries are at their lowest point in the fishing calendar, really, or the fisheries calendar at this point in time, and we can only achieve a full assessment of the health of the stock with full collaborative work with the fishing reps themselves. So we met at the end of February to talk through some of that and talk through what we were planning as an IFCA. Tim Smith's on the call. So Tim is my senior scientific officer. He's my scientific lead for the IFCA, and he will be leading and continues to lead on the technical side of that. So the next steps are there's multiple components to those. So we're accessing, or we have access to, historical information on catch and effort and landings. So an element is reviewing that and seeing where that sits as the fishing season progresses. We will also be, and officers are already active in doing this, so engaging with fishing vessels coming in. They do that generally anyway in their day-to-day work from the enforcement and compliance perspective, but gathering more information on what has been seen, what has been caught, the quantities, what fishermen are seeing from the different locations that they're working. We're really pushing hard on observer trips as well to supplement that. So getting out to sea, all of us, including myself. So I won't just be sitting in a nice cozy office that I'm sitting here at the minute. So it's for me to get actively involved in this as well around the coast. We're working some pots ourselves. It's a small number at the minute. Our normal pot and survey programme runs from May to September. So we're outside that at the minute, and weather is incredibly uncertain. This is really the first week, the first settled week of weather in a very long time to actually try and get gear work. So it's a smaller number of pots that we can be quite mobile with. Set them in different locations and recover them very quickly. And I want to put a huge caveat on Tim Robbins' open statement. We have just started this. We're at the early stages. We're really at the early stages. We're not in a position to say everything's okay. We don't know. We've just started. Initial signs in the first couple of weeks that we've been working are encouraging for lobster, but we've less information on other species such as velvet crab and edible crab. So the main species that we would be monitoring would be lobsters, edible crab, velvet crab, but also other benthic species that come up in the pots that fishermen are working. And then alongside that, we will be doing some camera work. Again, the time of year isn't particularly helpful because visibility isn't particularly great at springtime. So we will be progressing that as well as we go along. So that's kind of a broad picture of what we have planned. I'm planning regular meetings with this fishing group to update on what we're seeing and whether we can improve our monitoring programme. That information will also be shared across all colleagues on this call as well to draw in any additional expertise and support that can be provided to enhance that programme. Internally, our governance is provided by a science advisory group. That group will meet regularly, monthly, I'm looking at the minute, to review and support our programme going forward. That will then feed into the executive and the authorities. And the plan is to share to communicate as much as we can, to share what we're finding and how things are progressing. But really, until the full season has unfolded, I think only then will we get a true picture and understanding of, where the fishery is sitting. So that's quite a trip through what we've got on. As I say, Tim is on the call. Tim, have you anything more to add to that?
Just that, shellfish health is something that we've not routinely monitored, so we don't hold any data. I mean, when we are surveying, we will record black spot on crab, but there's nothing else that we would record in terms of the health of animals. Obviously, anything we see coming in on the quayside is alive and normally fine. So one of the things we're doing is collecting this data on shellfish health. So when officers are off on commercial vessels like today, or the surveys that we're undertaking for our patrol vessel, we are collecting information on if there are any more symptomatic animals, and then if there are any dead animals, because at the moment it's not routinely collected as part of our program.
Yeah, and I'm anticipating our first kind of update, and I'd provided some provisional dates for next meetup, and our first update probably around the 12th, 13th of April. So I've put those dates out, and that's for the fishing group, and we'll be in a position hopefully to present a bit of information and a bit of data on how things are progressing. But there are massive caveats at this point in time, and I just can't emphasize that enough, that we are just starting this work. And I'm really encouraged by the first meeting we had with, as a collective, the IFCA and the fishing group, and I'm really hoping, well, not hoping, I know we can move forward really collaboratively and effectively in co-production with that. Sorry, buzzwords just creep out, but you know what I mean. So thank you. Thanks, Tim. I'll take questions if anybody has any questions on that.
Okay, thanks, David. So are there any questions to David at the moment on next steps that the IFCA are undertaking? Sorry, Sally, yeah.
Yeah, I just wanted to add, because obviously my perspective is not sort of the fishing industry as such. It's obviously the marine mammals that I believe have been impacted. I mean, do you want me to try and collate some sort of formal data? Because we're still now regularly having seal pups which are coming ashore who are their birth weight and only just slightly more than their birth weight. I mean, they should be weaned off end of December at 30 kilos. And we're picking 4 or 5 up a week who are weighing between 14 and 17 kilos with no other health issues other than they're emaciated. And that tells me they're starving to death. And obviously, when first weaned off, they will basically scavenge the seabed, because chasing fish is a skill that they learn. And so they will pick whatever's up on the seabed, and clearly there isn't anything much for them to eat. So do you want me to try and collate figures on previous years? Because the zone that's affected stretches from probably, I don't know, south of Whitby up to sort of Seaham is the area which has particularly been impacted, and this has not been seen at other colonies around the coast. And I've taken away Storm Arwen out of the figures, so we can't blame the storms for having caused this issue. This is purely starving seal pups that are basically not thriving and not putting weight on because they're finding nothing to eat. And we're also finding ones that we have dealt with, which we've marked, are then travelling greater distances than normal. They'll maybe be released at Redcar and they'll tip up 18 hours later up on Northumberland's border with Scotland. So they clearly, normally they wouldn't travel such big distances, and they'll only be doing that looking for food. And in that process of travelling as well, they've lost weight, so they're not feeding en route. So it is bigger than just the fishing industry. This is an entire ecosystem that's suffering, and that's where I come from. So I'd like to be able to input as much data as I can to you, and maybe we should be sampling the dead seals as well. I mean, we've had literally 100 at least that have starved to death, seal pups. You know, we've got fishermen telling me that adult seals are following the fishing boats 20 miles out, circling boats all day long. So clearly there's issues in the inshore waters. So yeah, so that's me. I'll shut up now.
No, thanks for that, Sally. That's appreciated. Joe? Okay, thank you, Jerry. Are there any other points at all that want to be raised at the moment from the industry side? Sorry, Paul, you're on mute, Paul.
There you are.
Yeah,
I'm
there
new
you go.
to these machines. Just like to say, there's the lads still working still trying to work the tyres up the T's, and they're seeing nothing. There's no velvets, no green crab. We've tried a few pots in the dock and Hartlepool, nothing. I was talking to one of the lads who puts a couple of pots in the dock. He says he hasn't seen a lobster since well before Christmas. We've been to sea twice and we've yet to see a velvet. Odd lobster, odd crab, but that's about it. When we're getting out the back of the six, we were down there last week. I think we had 160 a crab, and I think there was 12.7 a lobster. We turned 350 pots off for that, which is abysmal. That was on a fortnight lay. So that's all I've got to say, I think.
Okay, thanks, Paul. David?
Yeah, just for the colleagues on the call that aren't aware of what Paul was talking about there about the tyres in the Tees estuary, it tends to be recreational fishermen set used car tyres in the estuary to gather generally molting crab and it's all shore crab, but we have estimated there's probably about 2,000 of these tyres located inside the Tees estuary across quite a wide area. So that's what Paul's referring to there. So they set these tyres, and the tyres act as traps or places where the crabs can harbour when they're molting, etc. They're in that process. So yeah, it was just to add to that. But the work that we have just started will hopefully inform a position of where we think, to try and better inform a position of where the fishery is sitting. But yeah, just started. Thank you.
Okay, thank you, David. Are there any more, Sally?
Promise this is the last thing. I know you sound like you've pretty much underlined dredging and put that one to bed, but I've just taken receipt of 400 pages of reports, which I'm ploughing through. And believe you me, dredging hasn't gone away, and we've got 2 million tonnes about to be dumped. And I've looked at the results that were done by, is it Fugro, who were going to build the plant there? And I tell you what, that sediment is toxic, and it exceeds level 2 on many of the testing, which clearly states it should not be dumped at sea, and as far as I'm aware, that's where it's going. So please do not rule out dredging, because I have found enough evidence to show it's of major concern. So I cannot understand why nobody else seems to be finding it. It's there, and it's not even scientific knowledge that's needed to analyze it. It's common sense. It's there. If you just read it honestly, please don't rule it out. Please don't. Thank you. I'll shut up. I'm gone.
Right. Thank you for that, Sally. And obviously, we're very happy to have discussions around, future dredging that's going to be happening in the port as well. If you'd like to take that with either the EA on the testing side or the MMO on the licensing, we're happy to have that conversation going forward. But for now, just looking at the work we're looking at here and the evidence we found here, that's the reporting we've done this morning.
Mm.
Okay, are there any other points that anybody wishes to raise on the meeting this morning, please? Do you want to raise them now? Or if there's anything you feel that hasn't been answered during the call this morning, then very happy to go over them. Chris?
I was only going to ask Sally a question actually about the seals in particular, and whether is the Animal and Plant Health Agency involved in any way? Have they been sent to any other laboratory for analysis? The reason I asked that question, going back to the algal toxins, now again, don't know whether they could impact, but they do bioaccumulate through the trophic levels. So we are working with, obviously there's been seabird deaths reported as well, and the Centre for Ecology and Hydrology are obviously investigating that. Now, I've just found out that we have got other samples from the seabirds that are running through our lab to look for DSP toxins and whether they could be implicated in those seabird deaths. Now, again, I don't want to kind of, say that this is the cause, but I do think it's important. Obviously, if there's a lack of food, that's going to have an impact, but it would be good to know whether any of those animals the seals have been sent anywhere, because I know there was some work done on St Andrews as well on algal toxins in the past with seals.
Yeah, unfortunately, seals tend to get neglected a bit in terms of testing. Obviously, CSIP are all over any cetaceans that we have come in. And I did submit one a couple of weeks ago for asking them to do toxicology testing. So we've basically had 5 porpoises in a 2-week period have washed up around the Redcar area, which is sort of quite unheard of. 8 in total going down to Scarborough. So I think they may be looked at for toxicology, but this is what we're up against. Even getting cetaceans tested for anything other than a basic cause of death is really difficult. And certainly the seals, because what happens is the bulk of the seal pups that we deal with are put down by a vet because the class has been of a terminal weight and with no rehab spot for them. So they'll be put to sleep on humane grounds because basically they're not going to survive in the wild. And because they've been euthanized, they're then incinerated because obviously their system is then awash with toxic chemicals. So other than finding ones that wash up, and then we could potentially submit those, although quite often they take quite a while to wash up. They're usually missing a head, and I mean, I don't know how fresh they would need to be. But the problem is that they're all majority euthanized, which then I think would impact on any testing. Because I know even if we've sprayed the porpoises with agricultural marker, CSIP then won't test them for toxicology. So I mean, I'll certainly ask that question. Sorry, who was the agency again?
Well, I mean, I could be wrong here, but generally speaking, I would kind of I would potentially, but perhaps not. So yeah. I tell you what, I think the best thing to do, Sally, if you don't mind, can we take a conversation offline? Because I don't know, I can't promise anything here. But as I said, our lab are doing more samples on the birds. But I mean, there's something maybe we could look at together. Yeah. But we'll take it offline maybe.
Lovely, thank you.
All right.
Okay, thanks. Joe, did you want to come back in on questions, please?
Yeah, so there's a couple of things there, Joe. It was the first question I asked from Marine Lab as well about sediment, because it was something that was playing on my mind. Could they be identifying sediment plumes or sediment in the water? So we looked at rainfall. Obviously, dredging was considered as well. So I was told by Plymouth Marine Lab that for some of the algorithms, and certainly the platform, which is the S3 platform, which does show an algal bloom, albeit at lower levels, that does correct for sediment. They were confident about that. I think your question probably was more is about the kind of composite images, but I go back to the point that nothing's ever going to be definitive in the satellite data. That's not what we're hanging everything on in this case with the algal bloom theory. It's the other information as well. So we know that these harmful algal blooms were in the region at the time. We know the temperature was about that at the time. We also have the algal toxins data as well. So the weight of evidence there it kind of comes from other areas, not just the satellite data. But I mean, completely recognise the point that you're never going to be completely certain with satellite data. You know, you can't be. Yeah, again, I think it depends on what we'd be looking at. So, you know, I think if it was an oxygen crash, like we've already said, I think, you know, if it was that, it would be over a relatively short period of time. But the real unknowns here are around the toxins levels and what that can do to crabs. So, and we found that across that area. So, yeah, unfortunately, I'm in a bit of a pretty rubbish situation here where it's kind of just theoretical and we can't pin it down to anything. But you know, that's the nature of the beast, I'm afraid.
Thanks. I got pinged for being on mute for a change. Okay, thanks. Yeah, we'll get a written response back on the questions because we've got those from you guys already. So sorry we didn't have time to fully cover those this morning. Okay, then I hope you found that useful. I hope you found that the presentations were useful and the ability to have a chat, and maybe we'll have some further conversations offline as well on some of your specific questions. So what I would say is, thank you very much for attending this morning. Thank you very much for your help. Thank you for your help you'll be giving Dave and the team there at North East NIFCA as we look at sampling and monitoring going forward. And just thanks to everybody for attending this morning and for being so open and positive in the conversations. And also, it's the first time I've been on a call in a long time where there have been 3 Tims on the same call, which is unusual already. So thank you very much for your time.
Thanks, everyone.
Yeah, thank you. Thanks, everybody.
Thanks, everyone.
Bye.
Thank you. Bye.
Thank you.